While searching for a kidney donor, a Boston woman named Karen Keegan stumbled upon a mystery. When her three adult sons underwent genetics tests to determine whether they were a match for a kidney donation, the test showed that two of them weren't actually her sons. Keegan knew she was her son's mother; she had conceived and given birth to them. Figuring there must have been an error, the doctor pursued further, only to uncover something even more confusing. She was her children's biological aunt. It turned out that Keegan had a second genome in and tissues in the body. In other words, some of her cells had a completely different set of genes from the others. The second set of genes belonged to her twin sister, who had never been born. This condition, where an individual has two genomes present in tissues of their body, is called chimerism. The names come from Greek mythology, where chimera is an amalgam of three different animals. Individuals with chimerism might have two tones of skin or two different color eyes, but most are believed to have no visible signs of the condition. Chimerism can come from a twin in utero from a tissue or organ transplant, or happen between the fetus and a pregnant woman. So how exactly does it happen? One of the most common forms a mother and fetus swap cells in the flows of nutrients across the placenta. The mother can't inherit fetal stem cells and differentiated cells that are able to develop into any specialized cells. The fetal cells initially go undetected because the mother immune system is suppressed during pregnancy. But in some cases, the cell with the fetus DNA persists in the mother's body for years or even decades, without being destroyed by her immune system. In one case, a mother's liver was failing, but suddenly started to regenerate itself. Her doctor biopsied her liver, found DNA in the regenerated tissue from a pregnancy almost 20 years earlier. The fetal stem cells had lodged in her liver and specialized as liver cells. Karen Keegan meanwhile acquired her second genome before she was born. Very early in her own mother pregnancy with her, Keegan had been fraternal twin. Keegan embryo observed some fetal stem system from her twin's embryo, which did not develop stem. By the time Keegan fetus developed an immune system, it had cells and with each genome, and the immune system recognized both genomes as her body's own, so it didn't attack or destroy the cells with the second genome. We don't know how much of her body was composed of cells with this second gene that can vary from the organ to the another, and even between tissues with an organ. Within an organ, some might have no cells at all with the second genome, while others might have many. At least some of the egg-producing tissues and ovaries must have carried the second genome. Each time that she conceived, there would be no way to predict which genome would be involved, which is how two her children ended up with this of the woman who had never been born. This can also happen to father. In 2014, when ancestry testing determined was actually his baby biological uncle. Researchers discovered that 10% of the father's sperm carried a second genome from an embryonic twin. Cases like this challenge our perception of genetics, though there are very few documented cases of cham- chimerism from the embryonic twin. They're all amalgams to some extent, carrying around the different genetic codes of our gut bacteria and even our mitochondria. mitochondria. And given that 1 in 8 individual births started as twin pregnancies, there could be many more with more people with two genomes, and many more lessons to learn about the genes that make us who we are.